So now it's open, presumably. I can't hear you, Dominica, I don't think.
You hear me?
Yeah, okay.
All right. Okay, I'm gonna wait 1 minute more and then we'll be good to go. All right, so good morning, everyone. My name is Domenica Berardi. I'm a postdoc at Yale University, and I'm co-hosting this webinar series of January, the 8th one, together with Diana Pinto from the ISEP from Portugal. And I really want to thank you for joining this webinar. Although like the number of participants is low, I would say that this webinar will be available online, so everyone will be able to listen to this webinar also like in the future. And also, I would really recommend to connect with EMN Society online in our Twitter account, and also by contacting the MedSoc through the emails that you can find here in this slide. So I think we're good to start with this webinar series, and I'm very glad and happy to introduce Dr. Andy Finch from the Breast Cancer Institute in the UK, and Kia Sateri from Breast Cancer Institute and Primary University. And this webinar will be about oncogene-induced apoptosis, a tale of metabolic and energetic supply and demand. So I want to introduce the first speaker, Dr. Andy Finch. As I've said, I'm very glad to introduce Andy. He did just a brief background on Dr. Finch. He did his PhD at the Trinity College in Cambridge and postdoc at the UCSF in San Francisco, and then back in Cambridge in the Laboratory of Molecular Biology, where he actually developed interest from cancer metabolism. And so then later on, he set up his lab at the IGMM in Edinburgh. I was very privileged of being like visiting the laboratory for a few months, where he had a huge mass spec facility, and yeah, a huge mass spec facility. He is also authors of several publications. He has given his contribution in the metabolomics field a lot. He was part of the Scottish Metabolomics Network and is currently part of the London Metabolomics Network Society. So happy to introduce you, Andy, and I give you space now for your presentation.
Thanks very much, Domenica, for a kind invitation, and Diana as well, to speak, and a kind introduction. Can you hear me?
Yeah.
Okay, that's a good start. Okay, so what I'd like to speak about today, thank you everyone for joining and listening, and I hope you'll find this interesting. It's about a process called oncogene-induced apoptosis, and this is a tale of metabolic and energetic supply and demand. Now, we published most of the work that I'm going to talk about in late 2022. And so what I really wanted to do was give a more biological introduction, why we're doing the work, why we think it may be important, and then to give you some of the core aspects of that paper. Then I'll hand over to Keir, who's going to present some data that's a little newer, I would say, some of which is unpublished. So yes, we hope that you'll appreciate that work is unpublished. Now, let me begin. Okay, so this talk is primarily about an oncogene called MYC, and MYC is, sorry, just let me hide that panel. And MYC. MYC is a transcription factor. It has a partner MAX, and together they are transactivators of many genes. Actually, if MYC is overexpressed, it's usually thought to be over 10%, certainly probably 15% of all mRNA genes in the human genome. And it's also a potent activator of RNA polymerases 1 and 3. So that means transcription of ribosomal DNA into ribosomal RNA and transfer RNAs, as well as some other small RNAs. Now, it's been known for a long time that MYC overexpression drives cancer. My slides are slow to advance, so just bear with me. Okay, so the primary function of MYC in cells could probably be described as a promoter of cell growth and proliferation. Now, as I mentioned to you, it transactivates many genes. Ribosomal RNA, of course, is a key determinant of cell growth. New ribosomes are required for cells to grow. Other MYC targets include E2F, for instance, which is involved in the cell cycle. But really, there's a large biosynthetic program that MYC drives, of multiple biosynthetic programs that mean that it's a very potent oncogene. Now, of course, if we deregulate MYC, and Keir will show you nice data supporting this, we do know that in most cell contexts can promote cancer. Now, fortunately for you and me, that process doesn't end up running away, because at the same time as promoting this cell growth and proliferation, MYC also sensitizes cells to a process called apoptosis, which you may know is a form of programmed cell death. Now, that is a good thing for us, because it means at the same time that MYC is making cells divide, it's also essentially killing them off. And so the end result of this is not a runaway into cancer. You can have models where MYC is overactivated, animal models, for instance, and the incidence of cancer is actually relatively low, you would say. So it's clear that this process of MYC-induced apoptosis, which is the phrase I'll use throughout the talk, that this process of MYC-induced apoptosis is clearly tumor suppressive, and it acts as a barrier to MYC-induced tumorigenesis. Now, work in my lab primarily focuses on the molecular mechanisms through which MYC triggers apoptosis. We think that's important because in many cancers that are MYC-driven, and it's often thought the majority of cancers, over 50% of cancers, harbor amplified or deregulated MYC in some way, overactive MYC. But many of these MYC-driven cancers will evade the apoptotic machinery, so they'll bypass apoptosis, and then, of course, that leaves them in a very tumorigenic state. So we would like to understand this process. We might be able to restore apoptosis in MYC-driven cancers, or even in cancers that haven't necessarily evaded apoptosis in the ways that we normally know about. We may be able to enhance apoptosis in MYC-driven cancers, and therefore try to treat them. Now, MYC is a transcription factor, and so for many years there was a search for transcriptional targets of MYC. So these are genes transactivated by MYC that might mediate this apoptosis. And that search went on for decades, actually, and we never found very convincing candidates. So a few years ago, I came up with an alternative hypothesis that isn't really based on individual gene targets of MYC. And so this is the hypothesis here, that if MYC is strongly overactive, we know that drives growth and proliferation. These, by definition, essentially bring along metabolic demand. And so I hypothesized that it was an excessive metabolic demand that actually underpinned the induction of apoptosis. And so that is the framework within which we've worked in my group for several years now. So this has been one of the main model systems that we've used in the past. We look in primary human fibroblasts. It's very important when you're studying mechanisms of tumor suppression that you have cells that have a full genetic complement. So we can't use cancer cells that may have deleted tumor suppressor genes, for instance, because that could impact the machinery of apoptosis. So we look in primary human fibroblasts, we overexpress a switchable form of the MYC oncogene, and we can turn that on or off as desired. And so what you'll see here on the left-hand side is an experiment where in these primary human fibroblasts, MYC has been activated overnight, and then we've withdrawn glutamine. And what you can see from the bars here is withdrawal of glutamine is very well tolerated by cells, and by normal cells. I'll come on to that in a minute. This is the vector control. But when you're overexpressing MYC, glutamine withdrawal now is a very potent apoptotic trigger. So we often say that MYC sensitizes cells to apoptosis, but I think probably a better way to look at this is MYC can drive this apoptosis that can be strongly suppressed by glutamine. So glutamine withdrawal is a very potent trigger of this process, and the fact that glutamine was involved really started our journey into cancer metabolism. We felt that there was likely a metabolic mechanism underpinning this glutamine sensitivity. What you can see on the right is a slightly different apoptosis assay looking at caspase 3,7 activity, but here you can see it's only in conditions where glutamine is withdrawn, and the red line here is with MYC overexpression, that we get apoptosis, and it's very rapid, this trigger. It's starting at 3 hours, and by 6 hours, the process is well underway. So this is clearly too fast to be any kind of transcriptional response, again indicating that it's probably metabolic or homeostatic. Now, we know quite a lot about the biology of glutamine, and so this was a good place for us to start. This is the same cellular system, and what you can see here is a high-content microscopy experiment in which individual dots, individual pixels, if you like, are areas of transcription, what we call nascent transcription. And we've used a pulse of ethanol uridine, which here is called EU, to label transcription in real time. So what you can see in a plate of fibroblasts, this is very much zoomed out, which is why the dots are so small. There are thousands of cells in the image, hundreds to low thousands. And what you can see is there's a basal level of transcription. When we withdraw glutamine, you'll see from the color range, that transcription is now strongly suppressed. On the color bar, we've moved down close to 0. And that's a normal transcriptional response to withdrawal of an essential nutrient, glutamine. Now, transcription, of course, uses nucleotides. It takes energy to make nucleotides, and glutamine provides cells with energy. But equally importantly, glutamine is required for nucleotide biosynthesis. So it makes good sense that the cell shuts down transcription in the absence of a key precursor of nucleotides. Now, when we overexpress MYC, and this is in the presence of glutamine, we now get a strong enhancement of apoptosis. You can see the color range moving up towards the white there. And this is, if you compare vector with MYC, this is just driven by MYC alone, this amplification, if you like, or strong increase in transcription. But this is now where the problem arises, when we take away glutamine in the MYC-expressing cells. And what you'll notice here is that the color range is again around the middle. It looks very much like the vector control cells, which of course are growing in the presence of glutamine. So what we can see here is that MYC is driving a level of transcription, quite a high level of transcription, even in the absence of a key supply of a metabolite, an essential metabolite, glutamine. So we like to think of this as MYC overriding the normal cellular responses to withdrawal of a key nutrient, glutamine. And that, of course, raises the potential for some kind of metabolic crisis. And that's what we went on to explore. So given that what I've just shown you is about transcription, and transcription needs nucleotides, one of the first things we looked at was how does MYC-driven demand affect metabolism? And we focused on the 2 core carbon metabolic pathways of glycolysis and the pentose phosphate pathway. So both pathways have a number of outcomes, and for the pentose phosphate pathway, the primary 2 are the production of NADPH, which can then be used for lipid synthesis or redox protection from ROS, for instance, but also the generation of nucleotides, which we were particularly interested in. So I'll show you now some stable isotope tracing experiments interrogating these pathways. So this is a mass spectrometry experiment. It's stable isotope tracing using 13C6 glucose. That's glucose in which all 6 carbons are carbon 13. And biologically, that acts exactly like normal glucose, but on the mass spectrometer, we can tell it apart. So it's an excellent way for looking at metabolic fluxes. These are sort of pulse experiments. So if you look over towards the loop here on the right, this is an abbreviated pentose phosphate pathway. We have 6 carbons coming in from glucose to 6-phosphogluconate, at which point we have reactions that convert those with loss of 1 carbon to ribose 5-phosphate, which you'll be familiar with as a precursor of nucleotides. Now, we've used a trick in this experiment where we've employed 6-amino-nicotinamide, which will block this reaction here, the loss of the carbon from 6 to 5. And what that does, using 6-amino-nicotinamide, is it blocks the pathway and it lets us see accumulation of metabolites indicating metabolic flux. So here, if we look at 6-phosphogluconate, in the vector control, we have this very small amount of phosphogluconate with a tiny amount of the 6 carbons, which are here in orange. But now when we increase our dose of phosphogluconate, we're blocking the pathway, and you can see we're accumulating 6-phosphogluconate, and it's primarily coming from glucose with these 6 carbons. So this is a really nice way to reveal the flux through the pathway that you couldn't see without the 6-amino-nicotinamide. Now, when we look the other side of that reaction, we can see that ribose 5-phosphate, I'm just going to concentrate on the MYC cells here, ribose 5-phosphate is now beginning to diminish as we block that reaction, and that is expected. Here you can see the 5 carbons of ribose 5-phosphate coming from glucose through this route. But what you'll also notice is there's this very clear accumulation of 3 carbon units in ribose 5-phosphate. Now, these 3 carbon units are not coming from glucose because all 6 carbons are labeled. So that gives us the yellow 5 labeled ribose 5-phosphate. What this actually indicates, and this is one of the really nice things about stabilized-isotope tracing, is that we've got an alternative pathway of ribose 5-phosphate synthesis here. And that pathway is in fact coming down from glycolysis, and the activity of MYC is diverting carbon from glycolysis back into the pentose phosphate pathway. So we get what's essentially a reversal of the lower part of the pentose phosphate pathway to give us this ribose 5-phosphate. Now, why is that happening? Well, it's because of that demand for nucleotides that's driven by MYC and driven by that excessive transcription. So this is very much a really nice example of how, due to oncogene activation, a global process, so transcription, can drive alterations in metabolism. And I like to think of this as the process is pulling metabolites through metabolic pathways. So you can see that's the effect of MYC on these pathways. Now, I've shown you already that MYC can elicit changes in metabolism, quite drastic changes in metabolism in the previous slides. I've also shown you that MYC can override homeostatic responses to withdrawal of glutamine. And so we wanted to find out why glutamine suppresses MYC-induced apoptosis, or asked the other way, why when you take away glutamine is apoptosis triggered? So you may be familiar with the biochemistry of glutamine. Amongst other things, it supports the TCA cycle. This is one of its major functions. And the TCA cycle itself supports energy, of course, because we produce reducing equivalents that go into the electron transport chain and oxidative phosphorylation. There are also several metabolites that would be considered biosynthetic that emerge from the TCA cycle. So this includes other amino acids. Fatty acids essentially come off from part of the TCA cycle. Heme synthesis is dependent on the TCA cycle, and various things that support nucleotide synthesis as well. So we can see this dual role, at the very least dual, of the TCA cycle in energy and biosynthesis. And you can also consider the fact that production of alpha-ketoglutarate, which can act through dioxygenases to promote various signaling pathways. So it's a really integral and pleiotropic cell pathway. So the first thing we did, again by mass spectrometry, is look at the impact of glutamine withdrawal, and this is whether MYC is present or not. What you'll see is that a number of key TCA cycle metabolites, alpha-ketoglutarate, fumarate, malate, and aspartate, essentially disappear when we withdraw glutamine. So we know that the effect on the TCA cycle of glutamine withdrawal is fairly profound. Now, other groups had been interested in similar questions, and of course, it's very difficult to dissociate by synthetic roles of the TCA cycle from energetic roles, because you're going to inhibit both when you take away glutamine. But we found something that we weren't expecting to find in the beginning, which was now in a much more MYC-dependent fashion, that when we withdrew glutamine, we were finding products of nucleotide catabolism. So in this case, nucleosides, so without the phosphate groups on them. What was particularly interesting about this is nucleotide catabolism the appearance of these species is really diagnostic of catabolism. So when you make nucleotides, they already have the phosphates on them. You don't see free nucleosides or free bases in these cases in normal biosynthetic metabolism. So these have been broken down. And actually, when you look down at the very end of the purine catabolism pathway, what we saw, again, only in the condition of MYC with glutamine withdrawal, we began to see a steady accumulation of xanthine. So it was clear to us that under under conditions of apoptosis, we were getting this nucleotide catabolism. And it's been known for a long time that in things like exercising skeletal muscle, if you deprive skeletal muscle of nutrients, in other words, you put it in an energy poor state, this is where you start to see products of nucleotide catabolism, particularly xanthine and hyperxanthine. So we felt at this point that what we were likely seeing was some kind of energetic stress. So we looked at our cellular energy charge. When you just focus on ATP, you really don't see a major effect. And I think that reflects the need of the cell to maintain ATP at all costs. But where you do now see quite profound nucleotide changes is in the lower energy adenine phosphate. So AMP, adenosine monophosphate, you can see this accumulation under the apoptotic condition, the same with ADP. And if we now use a ratio of the peak intensities of ATP and AMP, and this is on a log 10 scale, what you can see is in the apoptotic condition, we have essentially a crash in our cellular energy charge. Now, Kia will talk to you a little bit about AMP kinase, but there is a kinase in the cells that responds to states of low energy, and the response is it can be measured, if you like, by its phosphorylation on a threonine 172. So here we can see under our conditions, vector and MYC, glutamine present or absent. It's under the MYC condition with glutamine absent that we now have this signature of low cell energy charge. So at this point, we were fairly confident that we were getting a situation of low energy charge that correlated with the apoptotic condition. But what we really wanted to do was do some sort of perturbation experiments to get more mechanistic insight. And as I've said to you, that's very difficult to do with the TCA cycle because it's very difficult to perturb just the energetics of the TCA cycle or just the biosynthesis. The two go hand in hand. So we focused on a kinase that's present in the intermembrane space of the mitochondrion. It's a member of the adenylate kinase family, of which there are 9 family members. And AK2, as I'll call this family member, it plays an important role in facilitating mitochondrial ATP production. So if you imagine in the matrix where we have our ATP synthase generating ATP from ADP, that ATP now needs to leave the mitochondrion to be usable for the cell. It comes out through this transporter called the adenine nucleotide transporter into the intermembrane space and onto the cytosol. But in this space, AK2 will use some of that ATP, also mopping up AMP, and we know we have a lot of AMP, to produce more ADP to keep this cycle running as efficiently as possible. So in this way, AK2 actually facilitates overall cellular ATP production. So this is not to do with carbon, it's not to do with the TCA cycle, and we thought by perturbing AK2, we might be able to see if we could perturb the energetics, and by making the energy charge worse, whether that promoted the apoptosis, or by making it better, whether that inhibited. So here you can see a situation where we've knocked down AK2. So that should make the efficiency of the mitochondrial energetic machinery much lower. And so what I've written here is reducing energetic supply. What you'll see in the ATP-AMP ratio is with the vector control or with the MYC, which is in white, that when we have siRNA to AK2, we get a drop in energy charge. Now, as I showed you before, take away glutamine, and we end up with this strongly, it's a negative log 10 ratio. And once again, in the absence of AK2, the energy charge is further depleted. So we are confident that our perturbation of AK2 is doing what we think. Taking it away is reducing our energetic supply. And sure enough, consistent with our hypothesis, when we measure percent of apoptosis, this is all in the absence of glutamine, you can now see that knocking down AK2, reducing energetic supply, does quite markedly enhance the apoptosis that's promoted by MYC. Similarly, we did the opposite experiment. We overexpressed AK2, which will be in the intermembrane space, and what that we hypothesized would do would improve the cellular energy charge. More AK2 should make more efficient nucleotide transfer. Here you can see mRNA expression in our system. We got several fold overexpression, and you could see by Western blot that we had an enhanced expression of AK2. And so under those circumstances, we expected to see a reduction in apoptosis, and that's exactly what we saw. Under the conditions of MYC with glutamine withdrawal, we now get a marked reduction in apoptosis. So it does look at this point as though it is all to do with mitochondrial energy production, and that is the role that glutamine plays, is to support mitochondrial energy production and thereby inhibit apoptosis. So there's a lot more detail, some of which Kia will go into, supporting the energetic status of this process. But one of the things that we do, we strive to do as cancer researchers, is to move things towards some sort of clinical application. And so we wanted to see, now that we'd managed to pick some of these things apart, whether we could gain any therapeutic benefit from interfering with some of this machinery. Now, it's been known for some years that MYC overexpressing cells are glutamine dependent. And so there was a lot of interest in glutamine metabolism in MYC-driven tumors. And there was a target, glutaminase 1, that had a number of drugs were raised against by different companies and went into clinical trials. And here's two of them here as examples, BIPTES and CB839. Now, the metabolism of glutamine was very exciting in the MYC field, I think. But unfortunately, those compounds were not raised against the ideal target, I would say, because many other enzymes convert glutamine to glutamate other than glutaminase 1. And in fact, in clinical trials, the glutaminase inhibition has really been pretty ineffective. But I think there's a phrase we use that you don't want to throw the baby out with the bathwater. So if we know glutamine metabolism is important in MYC-driven systems, and so we wanted to see if there was another area where we could better target the metabolism. So we focused on this reaction here that's catabolized by OGDH. In contrast to the reaction catabolized by glutaminase 1, this has a very low level of redundancy. So there are very few enzymes, if any, really, that will convert alpha-ketoglutarate into succinyl-CoA. And so there's an inhibitor of this drug, an inhibitor of this enzyme, a drug called CPI613. And so we sought to see whether that might be a therapeutic target for us. So in collaboration with Ingo Ringshausen from the University of Cambridge, we used this drug in a MYC-driven lymphoma mouse model. So what this experiment is that we subcutaneously injected lymphoma cells into the flanks of mice. We then let lymphomagenesis occur, so tumors formed. But during the process of tumor formation, we treated daily with CPI613, this inhibitor of OGDH. And what you can see is we ended up with a statistically significant therapeutic benefit. So at the end of the experiment, when we weighed the tumors, they were clearly lighter, if you like, less mass in the CPI613 treated mice. Not only that, but the actual tumor area, what I should say really is the cancer cell area within the tumors based on histological analysis was also reduced. And so this was our first lead into therapeutic experiments, and we've got several more in design that we hope will be even more efficacious. So I'll hand over to Kia in a couple of minutes, just the conclusions. This is the paper that we published in 22. Really, one of the main conclusions is the title of the paper. So MYC sensitizes cells to apoptosis by driving energetic demand. And so it's important that we've got that distinction between energy and biosynthesis. I think the main three conclusions that I would draw from the paper are MYC overrides homeostatic responses, and that drives excessive metabolic or energetic demand. Glutamine protects from apoptosis by acting as an energy source, a mitochondrial energy source to inhibit the process. And targeting energy metabolism can be tumor suppressive in MYC-driven cancers. And I think this is important because I'm hoping we will see a resurgence of targeting metabolism, if downstream of glutamine, if you like, but just in terms of these cellular pathways, metabolic pathways. And I think that is likely to be an efficacious route to go down. There are a number of other smaller conclusions in the paper. If you're interested, please do read. And in terms of acknowledgements, there are many, many people who should be acknowledged. I've just picked out the main few here. Joy Edwards Hicks and Hu Zhong Su are joint first authors on that manuscript. They did a huge amount of this work, and we were very strongly supported in Edinburgh by Jimmy Wills, who was our mass spectrometry manager. The mouse experiments were done in collaboration with Ingo Ringshausen at the University of Cambridge, and I'd particularly like to thank Maurizio and Jing Yu from his group, who helped us extensively. And then at Bart's Cancer Institute, where the work was finished, there's Kia, who you'll hear from in a minute, and Kubra Jonathan as well. So thank you very much for your attention, and I'll hand over to Kia now.
Thank you very much, Nick. So I'll hand this over to Diana so that she can introduce Kia as well.
Thank you, Domenica.
Yeah.
Thank you. Thank you. First, thank you so much, Dr. Andy, for your presentation. And now I would like to introduce Kia Sateri. Kia studied biochemistry at the University of Portsmouth and did his master in biotech at the University of Birmingham. He joined Dr. Andy Finch's lab in September 2020 to study metabolic mechanisms of oncogene-induced tumor suppression. Thank you, Kia, for accepting our invitation, and the floor is yours.
Thank you very much for the introduction, Diana. I'm going to share my screen. Hopefully everyone can see.
Yes, yes.
Thank you for the introduction. I'm just going to be presenting sort of a short continuation on the paper that Andy just presented, mainly with a focus on looking at alternative ways that we can exploit the energy imbalance that's brought about by MYC to induce apoptosis in MYC overexpressing cells. So obviously, as Andy's shown, we already know that MYC causes this vulnerability to glutamine deficiency, and MYC overexpressing cells have this dependency on glutamine. Andy showed that intermediates of the TCA cycle are reduced when you withdraw glutamine, and of course, we get that apoptotic phenotype with the withdrawal of glutamine. But as Andy touched on, glutaminase inhibition hasn't been as successful as people would have hoped. So there's a lot of metabolic redundancy. There's a lot of genes that have a lot of other enzymes that have glutaminase domains that are highly expressed. And so because of that, we wanted to look at some alternatives for glutaminase inhibition to target glutamine metabolism in our MYC overexpressing cells. So as Andy touched on at the end of his presentation, CPI-613, which is an inhibitor of oxyglutarate dehydrogenase, was the main thing that was tried. He tried it in vivo, and this enzyme, OGDH, converts alpha-ketoglutarate to succinyl-CoA, so a little bit downstream of glutamine. It's a lipoA analog drug, so it inhibits OGDH with competitive inhibition, and it's an alternative to depleting glutamine, but it also targets pyruvate dehydrogenase, which also uses lipoA as a cofactor. So we can see that in our MRC5 fibroblasts, in the MYC overexpressing cells, we get a very similar kinetics of death and apoptosis, and we have very little effect on our vector control MRC5s with this drug. This is in vitro data, and we have some confidence that this is due to inhibition of OGDH in particular, because when we add a downstream product, succinate, we can rescue this apoptosis after 24 hours. So Andy's already covered this drug. We wondered if there's anything better that we can do. Are there better ways to target the TCA cycle? And that led us to look at succinate dehydrogenase. Succinate dehydrogenase converts succinate to fumarate. And we have a more specific potent inhibitor of that, which is at penNA5. And the nice thing about targeting succinate dehydrogenase is it's unlikely that there would be much redundancy here, because it's an enzyme that's also a part of the electron transport chain as a part of complex 2. So not likely to be as many ways of redundancy and overcoming inhibition of SDH. So when we look, when we treat our cells with atpenin A5, the inhibitor of SDH, we see again very similar kinetics, rapid apoptosis in MYC overexpressing cells with minimal effect on vector control cells. And this is statistically significant. So it shows that we can target this energy supply coming from glutamine in many different ways. And there are alternatives to glutaminase inhibition. But we thought, what about not looking at this glutamine-derived carbon? Are there any other ways that we could do this? And so, as Andy showed before, we know that overexpression of MYC deteriorates the ATP to AMP ratio on its own. And then when we take away that energy supply from glutamine, this is exacerbated and it's even worse. And in this condition, we get apoptosis. So we thought, is there any other ways that we could deteriorate this ATP to AMP ratio, cut off energy supply, or affect energy demand to target this energy imbalance? And naturally, that led us to look at AMPK. So AMPK is a master regulator of energy homeostasis in the cell. As Andy showed, we do have phosphorylation of AMPK when our cellular energetics are deteriorating, with MYC upregulated and with glutamine withdrawal. And usually what happens is when you get this low ATP to AMP ratio, AMPK is phosphorylated, it's activated, and the net effect of this is that we get reduced energy demand by shutting down fatty acid synthesis, protein synthesis, glycogen synthesis, and bringing about new energy supply by upregulating glycolysis, glucose uptake, and mitochondrial biogenesis. So we thought, what ways could we have to target AMPK? Of course, we could inhibit it. So we use this inhibitor dorsomorphin, and of course, we can activate it. So there's two other ways that we can look at this, and we can see the effect that this has on our cells that are overexpressing MYC. So first off, we looked at this inhibitor dorsomorphin, which is an ATP competitive AMPK inhibitor. And what we found is even in the presence of glutamine, it was sufficient to induce apoptosis in our MYC overexpressing cells exclusively. So it has no effect on the vector control cells. But we see very rapid, at 9 hours already, quite rapid apoptosis in the MYC overexpressing cells. So then we thought, what if we do the inverse? As I said, there are activators to AMPK. What if we take a condition where we know that we have energy stress and that's causing apoptosis, so glutamine withdrawal, and then we use an activator of AMPK to try and alleviate that apoptosis? And what we found is it worked exactly as we expected. When we use an AMPK activator, we can rescue apoptosis from glutamine withdrawal at 24 hours. So we're fairly confident that through perturbing AMPK, we can target this energy imbalance. And then from there, naturally, we thought, are there any other ways, apart from direct targeting of AMPK, that we could affect this apoptosis? And that led us to AMPK-related kinase 5, or ARK5. The reason why it's called AMPK-related is due to its high sequence homology that it has with AMPK alpha, which is the catalytic active subunit of AMPK. And ARK5 is considered to be part of that AMPK alpha family of kinases. And previously, Daniel Murphy's group in Glasgow had shown that overexpression of MYC seems to induce some sort of reliance on ARK5. And they thought this was happening via upstream regulation of AMPK. So we decided to test this in our cells to see, through targeting ARK5, could we also recapitulate that same phenotype as when we inhibit AMPK? So as we know, when we have MYC up and we remove glutamine, we get this deterioration in the ATP to AMP ratio. This activates AMPK. We've already shown that we can target AMPK. If there is this regulatory aspect upstream via ARK5, then if we inhibit ARK5, it's possible that we can then get to the same phenotype in the end. And we decided to use HTH01015. And one of the benefits here is this is a highly specific inhibitor that's been tested in panels of different enzymes, including other members of the AMPK alpha family, and it has very little effect on them. So we decided to have a look at this and see if we could inhibit ARC5 in our cells and see the impact that that has on apoptosis. So what we found is, in the absence of glutamine in our energy-stressed condition, ARC5 inhibition accelerates the apoptosis from glutamine withdrawal. And that suggests that it is part of orchestrating that homeostatic response to the energy stress that's brought about by MYC overexpression and by glutamine withdrawal. And then what we found was, again, as with dorsomorphin, our AMPK inhibitor, just inhibiting ARC5, even in the presence of glutamine, is sufficient to eventually induce apoptosis exclusively in the MYC overexpressing cells. So we were very happy with this result. We decided then we need to check to see if that regulatory pathway with AMPK that was reported by the Murphy group is in play in our cells. So we decided to look at phosphorylation of AMPK and phosphorylation of a downstream target of AMPK, which is acetyl-CoA carboxylase. Now, ACC catalyzes the very first step of fatty acid synthesis. So usually AMPK, once it's phosphorylated, phosphorylates ACC, and it shuts down that very energy-intensive fatty acid synthesis. So what we find is, as Andy's shown before, when we take away glutamine, we get an increase in phospho-AMPK and an increase in phospho-ACC. But when we use this inhibitor of ARC5, we actually have less phosphorylation of AMPK and we have less phosphorylation of ACC. So it very much is possible that ARC5 can regulate AMPK phosphorylation and thus affect our apoptosis that way. So lastly, I'm just going to touch on some of the current work that we're doing, which is to test in B-cell lymphoma. Andy showed a mouse experiment with a model of Burkitt lymphoma. And we think lymphoma is a very useful model for us to take our work and make it more translational, because as you can see, in non-Hodgkin lymphomas, there's a very high frequency of MYC mutation. And the nature of these mutations is often translocations of the MYC gene into a highly active locus, which is the IgG locus. And in addition to that, in lymphomas, you often get translocations of BCL2, and the BCL family are the primary regulators of apoptosis in cells. So BCL2 overexpression tends to prevent apoptosis. It's the primary anti-apoptotic gene that is mutated. So we thought that this would be a perfect model for us to look. And what we decided to do is characterize the levels of MYC and BCL2 in a panel of different cell lines that we had, primarily diffuse large B-cell lymphoma lines, as well as a few Burkitt lymphoma lines. So as you can see, we have some cell lines that are high MYC, some that are high BCL2. So what we decided to do is take U2932, which is a high MYC line, and SUDHL2, which is a low MYC line. And we decided to have a look at some of our inhibitors to see if we could induce apoptosis in that MYC-dependent way that we've shown in our previous models using the MYC-ER fusion protein. So what we found is, after 48 hours treatment with dorsomorphin, which is our AMPK inhibitor, specifically in the higher MYC level cells, we get a doubling of apoptosis from baseline. And it has no effect on SUDHL2, which is a cell line with very low MYC. And so our current work is to test various concentrations and combinations of atpenin A5, dorsomorphin, HTH01015, as well as venetoclax, an inhibitor of BCL2, to see if we can find a therapeutic window in which high MYC lymphoma lines respond and low MYC lymphoma lines are unaffected. And we want to also test these on normal human B cells to see if there are any off-target effects. And that's currently where we are with this project. So I'd like to thank several groups at Barts Cancer Institute that have helped us with this work. Of course, the TCM group, Tyson Sharp group, and John Richards group, as well as the mass spec facility in Edinburgh. Thank you.
you very much, Kia, and thank you very much, Dr. Finch, for the very nice presentation, we both. I think if we have questions in the chat, we can have a look. And if not, I would like to actually start with a couple of questions that I have for you both. The first one is for Andy. And yeah, I was thinking, so the flux metabolomics was for intracellular metabolites, not for extracellular, or do you look also at the well, for example, potential ratio between the metabolites that have been internalized and the ones that have been extruded, like from the cells?
We haven't looked at that so much. We have a little bit, but in large part because most of where we're focused is a long way inside the cells. So it's mitochondrial, obviously. And so in combination with withdrawal of glutamine. It is an important question, actually, because the impact of glutamine withdrawal is, I've rather simplified there as being a loss of TCA cycle, but actually there are a lot of broader implications, I would say. So one of the ones that you can easily imagine is you get loss of glutamate. Glutamate, in addition to feeding the mitochondria, of course, it does a number of other things, which include activity on the cysteine antiporter, the XCT system of cysteine uptake, which is coupled with glutamate export. So you can immediately see that just withdrawal of glutamine is probably now going to impact your ability to uptake cysteine and various other things as well. So no, I've given you the reasons why we haven't really focused so much on things outside the cell. I think there is an importance of that export import. What we have really tried to do, though, is look more closely at export import into and out of the mitochondrion. And so one of the ways we've done that, which actually we've used this technique in the paper that you're on, Domenica, from 2022, is to do a rapid fractionation, a rapid permeabilization technique to try and separate cytosolic from mitochondrial metabolites. And so it's to address the same sort of issues, but in terms of mitochondrial import and export.
Right. Okay. And the other question is about the in vivo studies that you've done. I was wondering, well, actually, like, glutamine is involved in so many other kind of pathways, not just nucleotides, but it goes into lipids, and then lipids are involved in hormone productions. And when you get down to hormones, things like the gender differences are a sort of obvious, like, thing that you might imagine could be relevant. So I was wondering, have you ever thought about also going into that kind of difference between males and females, and if, like, glutamine deprivation could have like any sort of different impact on also the expression of NIC in males compared to females?
We haven't addressed it, and we're not going to, partly because I'm sure you're right. I'm sure there's probably huge impact. You know, we often think, because you look at metabolic maps, we think we know what metabolism is going on, but I think that's very naive. And then when you bring things like hormones in and signaling, which will have knock-on effects, it's extremely complicated. So I do think glutamine deprivation, if you like, if you could achieve that pharmacologically, maybe through blockade of probably multiple transporters, would have quite profound effects. The reason I say we're not going to do it really is that we are trying to move our work into a more refined, and I don't mean that in a pretentious way, but to reduce the complexity that we have with glutamine withdrawal. So that's why Kia, for instance, spent a while talking about appennin A5, a very specific inhibitor. And so we're trying to really be more specific with the aspects of glutamine metabolism that we inhibit, both to hopefully enhance the effects of what we see, but also to reduce off-target effects like the ones that you've just mentioned.
Right, okay. Yeah, makes sense. No, thank you, Andy. And like I've got another question, sorry, like Diana, then you can go like if you have any questions. A question for Pia. You were showing the exposure to glutamine of your cells that have a high expression of MYC, and then also like the exposure to glutamine withdrawal. And I've seen in one of your graphs that for glutamine withdrawal, you had a time frame of 6 hours, and then for glutamine like addition, you have 24 hours. And I was wondering, why did you use that different time frame from glutamine withdrawal and then the addition of glutamine? And do you think that might have an impact then also on the expression of AMPK?
Well, the kinetics of death are different. So when I was showing, for example, acceleration of apoptosis after treatment with an ARC5 inhibitor, beyond, let's say, 9 hours, for example, it's just flatlined because all the cells are dead. So I limited it down to that time frame because that's when the apoptosis is occurring, and that's where you can see the acceleration. With the 24-hour time points in the presence of glutamine, it's because the kinetics of death are slower. So I'm simply choosing the cutoff point based on when I'm seeing the majority of the apoptosis and then the plateau of the death. With regards to whether it changes AMPK expression or whether phospho-AMPK is different at different time points, that is true. All of the plots that I showed were taken at a 6-hour time point, so very early on in the onset of apoptosis, because the time point was selected specifically to show in that particular state, right before the initiation of apoptosis, what the conditions in the cell were like. But with a time course, you can see more in terms of how this develops through time. Yeah.
Thank you. That clarifies.
Thank you. Domenica, I have some questions also. First for Dr. Andy Finch. I'm from a different metabolomics field, but as I understand, in the in vivo study, the route was subcutaneous, and I would like to ask, why was subcutaneous route selected, if it is a reason for it?
Yeah, so the tumor was subcutaneous, so the injection of the lymphoma cells. The drug itself was given IP, intraperitoneally, which is, usually leads to more rapid dissemination. The reason that we use subcutaneous lymphomagenesis is somewhat one of convenience. So there are probably a slightly nicer way to do it is tail vein injection, and you'll get lymphoma in the spleen. The difficulty is that's very hard to monitor, of course. And so what we wanted to be able to do was assess the rate of tumorigenesis, and then, of course, to sacrifice the mice at the proper humane endpoint. That's very difficult to do with an internal tumor system. So I think it is a slight limitation. It wouldn't be my preferred sort of ideal cancer biology experiment. But actually going forward, we will continue to use that, in part because we'll start, as we do more of these perturbations and more refined perturbations with drugs like appennin A5, we also want to see what's happening to the metabolism, for which we will do stable isotope tracing in vivo. And of course, if you're doing that, you need to harvest your tissue and get it frozen as quickly as you can. And so for those experiments, really subcutaneous for us is really the only realistic option. So it's with that in mind as well.
Okay, thank you. And also a more general question. What do you think it would be the next steps or future perspective on this work? What will you do next?
For the field in general, just let me start with that.
Yeah.
MIC has been a target for a long time, and it's been very difficult to drug directly. So, you know, there have been friends of mine actually and colleagues who have been trying to target MIC directly, and so in tumors that are MIC driven to reduce the levels of MIC. There are people who are looking at the transcriptional program activated by MIC and drugs that can interfere with that transcriptional program. So there are already people working in the MIC field that are taking different approaches. I think our approach, there aren't many people doing it, and I think part of that is what Kia and I both alluded to, the failure of these glutaminase drugs in clinic. Now, I think the reason for failure is fairly obvious, and I think in retrospect they weren't the best targets, but it's a thing of implication, I think. So people have steered clear again of this area. What I'm hoping our report will do from 2022 and the work that we're doing now is give a resurgence to the idea that you can target the altered metabolism in MIC-driven tumors. So it's still quite early. I hope that we can help that side of things grow, and we have colleagues who are interested in similar things, and we have actually a very good dialogue with them. So I can't really answer your question, Dana, but I hope this has some mileage in it. We'll find out.
Yes, I believe you will have good fruits from your research. And also, I have a question for Kia. Also, I'm a student, so I think I was thinking, what do you think is the most challenging task of your work that you have done so far?
That's an interesting question. I'd say the most challenging thing is just sticking to, or rather continuing to press on when you have difficulties with cells or things like that not working, or trying to continue on work that's previously been done with different systems, different equipment, different everything. And trying to recapitulate results was really the most difficult part. But once we got things going, I've had a pretty good time of it. So it's been good.
Okay, great. Thank you, Kia.
We did have some terrible trouble with cell lines, with the cells,
Yeah.
but
Yes,
that's not restricted to our group. That's most groups.
Oh no, I can say that it's for everyone.
Yes, for me too.
Yeah. No, really, thank you very much, Andy and Kia. That was like an amazing talk for both of you. And as I've said, like earlier, this webinar will be available on the online platform of the EMN Society. So everyone will be able to have access to it. And we'll actually promote this webinar also on our social media as well. Thank you again very much. And if there are no other questions, I think we can just end it here.
Thanks, Domenica. Thanks, Diana. It was a pleasure.
Thank you. Thank you so
Thank
much.
you. Thank you very much.
Thank you so much.
Thank
Bye,
you.
everyone.
Bye. See you.
Bye.